Well, good morning or good afternoon, depending upon what time zone you're in. I don't think we have any good evenings at the moment. I would just like to, first of all, thank Estelle and Marco for getting this all set up. Estelle particularly for all her past service in doing this, and Marco for all his current and future service in doing this. And I'm sure that we are going to continue to have a very technically successful as well as scientifically successful series of director seminars. I'm also really, really thrilled as the new director to be able to kick off this seminar series with a great talk by a great speaker. I'm particularly excited about what Julie is going to talk about, not only because it really is an important advance in our understanding of the microbial population of the oceans as a whole, but also because this piece of work links together two very important things. One is the timescales in particular that are important to astrobiology, that is evolutionary timescales. And it links those evolutionary timescales to the very short timescales that are of interest to NASA's Earth Science Program and global change. And we're going to hear about that from Julie. Julie got her PhD in oceanography from the University of Washington in 2004. That followed her bachelor's in 1998 in marine science from Eckerd College and a master's from the University of Washington in 2000. Her dissertation title at UW was Phylogenetic and Physiological Diversity of Subsea Floor Microbial Communities at Deep Sea Seamounts, and that has in fact continued to be her principal research interest. The work she's going to be talking about today was published recently in PNAS in a paper by Mitch Sogan and several other authors. Her talk today is co-authored by Mitch, Hillary Morrison, David Mark Welch, Sue Hughes, and I apologize if I got the pronunciation of Sue's name wrong, and Phil Neal. And her title is Microbial Diversity in the Deep Sea and the Underexplored Rare Biosphere. And without further ado, I turn it over to Julie.
All right. Thanks, Carl. I also want to thank you for the invitation to give this talk today, and I especially want to thank Estelle and Marco, the IT people here in Woods Hole. It's been a pretty traumatic last couple of days without internet or email, and I really appreciate all the efforts, and especially the National Academy, where I have a beautiful view of the ocean while I'm giving this talk. I also want to thank my co-authors here at the MBL, as Carl just mentioned, and I think I'll just jump right in from there. Let's see. So this is a brief outline of what I'm going to be talking about today. I'm going to give you an overview of our motivation for doing this work, and then spend a good bit of time talking about the approach that we use. This is a 454 tag sequencing approach, which I'll talk about. I'll also discuss sort of how we crunch our data, since it's a really integral part of understanding the system. I'll go through some results, discussion and implications, and as Carl mentioned, I'll talk about some future applications, especially as they relate to earth sciences at NASA. So we all know that we live on planet microbe, and that they're the primary engines of Earth's biosphere, that microbes mediate biogeochemical cycles that shape planetary habitability. We know that microbes were likely the only form of life for most of our biological history. And we know that microbial communities of untold diversity continue to dominate nearly every corner of our biosphere. So understanding microbial diversity and community structure, then, are key to understanding our biosphere, our habitability, and our history. I'm an oceanographer, so today I'm going to be focusing on microbes in the sea. And if we look at the relative abundance and productivity of marine life, we can see that the smallest class of organisms in the ocean, the prokaryotes, which are less than 3 micrometers in size, make up almost 82% of the biomass in the ocean. And in fact, they account for over 90% of the primary productivity in the sea. So if we actually take a look at what some seawater looks like, if you filter it and you stain it with a nucleic acid stain and take a look, this is sort of what you see. And in fact, these larger objects here are protists. These the bigger of the small green dots are bacteria, and these very small ones in the background are viruses. And in fact, almost any place you look in the ocean, you're going to get a picture like this. And the amount of biomass in the ocean that is microbial is absolutely tremendous. And there's, by some estimates, on an order of 10 to the 29th microbial cells in the ocean. So I'm getting...
Julie, I'm going to interrupt you for a second. Someone has an open mic. Yeah, you're getting feedback. Please, all the Polycom sites, mute your microphones. And Mike Fitzgerald, if you would mute the conference call number, that should take care of it. We have some open mics. Berkeley, I think your mic is open. Thanks. Okay, and Mike, if okay. Julie, you should be okay, but I hear someone coming in. So if everyone can double check and make sure you're muted, that'd be great. Go ahead, Julie.
When that yellow light.
Berkeley, the red light means you're muted.
Oh.
Okay. Yeah, I keep seeing Berkeley on my end. I don't know why.
It may be a software problem.
Okay. So, as I was saying, the amount of cells in the ocean is absolutely enormous, and it's on an order of somewhere around 10 to the 29th microbial cells. And if we take a look at microbial interactions in the ocean, this is just a schematic showing some of the sources or ways of primary productivity in the sea, from heterotrophy to photoautotrophy and chemoautotrophs. And while we have a pretty good sense of what's happening in the upper water column, where photosynthesis dominates, we have much less knowledge about what's happening in the deep sea, for example, in the bathypelagic pelagic and other zones. And in fact, when it comes to really microbial diversity, community structure, and how these populations change over time, everywhere in the ocean we're lacking a great deal of knowledge. So this lack of knowledge is really what motivated the formation of ICOM, which is the International Census of Marine Microbes. And the goal of ICOM is to report what is known, what is unknowable but knowable, and what may be unknowable about the diversity of marine microorganisms by the year 2010. And the goal of our particular ICOM project is to determine the range of genetic diversity and relative numbers of different microbial organisms at sampling sites throughout the world's ocean. So this project is headed up by Mitch here at the MBL and Jan de Leeuw, who is in the Netherlands, and it's supported through the Census of Marine Life program, which is funded by the Sloan Foundation. So over the past couple of years, a number of working groups have gotten together for ICOM and have put together, okay, if we're going to do the census, how are we going to do it? And they've determined that determining the number of different types of organisms and their community composition is an essential first step in the census. And they also decided that the metric for the census needs to be molecular. This is not to say there isn't a place for culturing in the census, but simply that it's unrealistic to think that we can catalog all of microbial life in the ocean using culturing alone. And this is just exemplified by, if we look simply at the bacterial known phylogenetic divisions over the last 15 years. This is a figure from Norm Pace's lab. You can see that in 2004, over half of the known bacterial phylogenetic divisions are represented only by sequences and no cultures. And so it's very important that we use a molecular metric. Obviously, the ocean is big, there's a lot of cells, and the census is a big job. Even if its most simple form, conventional technology will be insufficient, and it will clearly require the development of new computational tools, which I'll be talking about today. So we discussed some possible solutions, and of course the first one we talked about was the one many of us are familiar with, and that is DNA sequencing of either PCR amplicons or shotgun cloning or phosmids of some sort. But really this technology is still pretty expensive, depending on what your lab can put out. And that cost constraint also then constrains the size of our current surveys to less than about 1,000 sequences from an environment. And that really only captures a fraction of the community structure. In addition, because you're only looking at 1,000 clones, it can be difficult to detect underrepresented members of the community, and it's also still pretty labor intensive. Even if you have robots doing a lot of work for you, it can still be pretty tough. We also discussed using DNA microarrays, but really you're limited by what is spotted or printed on your array. We discussed a lot of other solutions, but what we came up with and what I'll be talking about today is a tag sequencing strategy because we believe it has greater throughput and reduced costs. So a number of investigators have examined using the small subunit ribosomal RNA for tag sequencing strategies, and they focus mainly on hypervariable regions of the ribosomal structure, and those are shown here in red. So this is the secondary structure, and in it we have both conserved regions and variable regions, and based on our own experience in our lab, we chose to target this V6 region of the ribosomal gene. And so this, by looking at one part of the gene, we can infer taxonomic identity. So we designed primers, which are shown here in yellow, that are just to the highly conserved bacterial group. We didn't even look at archaea in our initial experiments. We then applied what I'm going to be explaining, which is called the 454 tag sequencing approach. And this is just a basic environmental DNA PCR reaction where you take your DNA from seawater or whatever sample you're interested in, and you amplify it with these V6 specific primers. But synthesized onto these primers are these life science adapters, and I'm going to explain why those are important in just a moment. So we perform a PCR with these primers, and what we end up with is a V6 amplicon library, and we sent that off to 454 Life Sciences, which is located in Connecticut. And what they give back to you is a number of tags from your different samples. And because this is a relatively new technology, and I know that quite a few people in the astrobiological community are actually using it, I thought I would just briefly review how this process works. It's also very different from traditional capillary DNA sequencing methods, so I also thought I would explain it. So these first two steps most of us are familiar with, where we extract environmental DNA, we amplify them with our specific primers of interest. And in a normal microbial ecology lab, from this step you would go on to cloning and Sanger sequencing. The 454 sequencing approach basically eliminates that cloning step. So what you have is your pool of DNA amplicons, your PCR amplicons, and these products are then denatured, and a single strand is recovered, and it's immobilized onto these very small beads that have a complementary primer to that A and B adapter that are synthesized to our own primers. And these beads are then deposited and put into an oil-water emulsification PCR, and that generates millions of copies of that single DNA strand on each bead. This reaction is then denatured, and they're put into something called a picotiter plate, and these plates were developed just for this technology. They have about, I think, 1.2 million wells in the picotiter plate, and each one is just big enough to fit one bead and your sequencing reaction reagents. Then solid phase pyrosequencing occurs. I'm not going to go through that in a lot of detail, but basically when a base is incorporated, a flash of light occurs. And in the end, you get about 200,000 reads per sequencing run, and their average length is about 100 base pairs, and the cost is somewhere around 2 or 5 cents per read. So remember, the goal of this is really to identify organisms in an environmental sample so we can do a census. So the key really is to identify the known universe, and we've spent a lot of time building a reference data set so we can understand what we're studying. So to do this, we collected full-length ribosomal sequences that contain this V6 region. We collected them from 4 major sources that are available freely on the internet. And then we put them through the RDP, the ribosomal database classifier, to get taxonomy. Many of them are from uncultured bacteria, have no affiliation, no taxonomy in the identifier. And from those known full-length sequences, we extracted just the V6. So what we were able to do is build a database that has a full-length ribosomal sequence with an associated V6 and a known taxonomy. And those are included in both a blastable format and a searchable SQL database. We then take our data through a number of different steps. And the first that I just want to focus on right now is this quality control. This is a very new technology, and we've spent a lot of time trying to understand exactly how to evaluate it in terms of being able to remove low-quality reads. It's important to note that this is very computationally expensive, and so we've had to develop a lot of new tools to properly evaluate this. And because of the general audience of my talk, I'm not going to spend a lot of time talking about the details, but we actually believe the error sequencing rate is quite low, and if anybody has any questions about that at the end, I'd be happy to answer them. We've done a number of control experiments remember that we're going into an environment where we don't really know what we're going to find. We can't benefit from, like when you sequence the genome and you get 10, 20, 30x coverage. So we're really trying to determine how well we can trust our data. So I guess I'll go through these two different pipelines when we get to the data, but first I want to talk to you about the pilot study that we performed for ICOM. And we collected samples from two sets of collaborators, and the first were our colleagues in the Netherlands. And these are Transat samples from cruises in the North Atlantic. The goal of these cruises is to track the formation of North Atlantic deepwater from its source through the oceanic conveyor belt. And these yellow and red dots are two cruise tracks they've had. And we chose just 3 sampling sites shown here in white. And from each of those sites, we chose both a sample from the oxygen minimum zone and also at greater depth in the ocean. For our second study site, we chose a very different deep sea setting, and that is my own interest, deep sea hydrothermal vents, in this case, Axial Seamount, which is located about 300 miles off the coast of Oregon on the Juan de Fuca Mid-Ocean Ridge Spreading Center. And from Axial, we chose 2 particular diffuse flow vents, and I'm just showing one of them here, which I'll be mentioning quite a bit because it's our largest data set. And what you're looking at, this is a diffuse flow vent, so there's warm fluids about 25 degrees C leaking out of the seafloor. You can see our sampler inside the vent trying to catch that flow, and a bunch of charismatic macrofauna around. You can see tube worms and limpets. This is some strange cilia that we don't really understand, and this white matter is microbial mats. So this is just a summary of those samples. You can see the paired Transat samples from the North Atlantic deepwater, each one from a different depth, and then the 2 diffuse flow samples, which are between 25 and 30 degrees Celsius. So the first part of our data processing, when we got this data back, I guess it was very, very early this year, I think it was 2 days after Christmas or something like that, was we took all of our data and we blasted it against our reference database. And you can use this side of the pipeline to get to the taxonomy. So you blast all of your sequences against the reference database, and then you take your query and you align it with the top 250 best scoring sequences using a program called Muscle. And from that, you can calculate the distances and identify the minimum distance, and that helps you get the closest identity in your reference database. You can also take that top blast hit and use that as sort of an estimate of diversity in your sample. So if you have 100 sequences and 80 of them blast to one sequence and 20 to the other, you can try to get a rough estimate of diversity. And that was the first thing we did simply because we didn't really know what else to do. And so this is the first result that we got back when we did that experiment. So these are rarefaction curves. For those of you who aren't familiar with them, they basically allow you to compare richness among samples that have been sampled unequally. And the curves result from basically averaging randomizations of the observed accumulation curve. And so these are not an actual measure of confidence about diversity in a sample, but they simply help you visualize how much more sampling needs to occur. And if we had actually sampled these environments to extinction, you would see these curves flattening out like this. But in fact, you can see they're still going way up. And we were very surprised by this result, simply because no one had really seen this in the marine environment. It's been discussed pretty thoroughly in the soil environment. But we were quite surprised, and especially since we knew this was an underestimate of diversity, because 2 very different sequences can blast to the same query. And so we decided we were going to have to do something a little bit more rigorous and not so naive, really. And in fact, that was backed up when we looked at how many blast hits we got for each sample compared to how many unique tags were actually in each sample. And you can see that there are many more unique tags than there are blast hits. And so we knew we were underestimating diversity using this approach. So the second time around, when we decided to do this a little more rigorously, we used a program called DOTR, which I'm going to explain. So we take all of our sequences, we align them, we calculate distances, and we pop them into this program called DOTR. And this was developed by Patrick Schloss in Joe Handelsman's lab. He's now at UMass Amherst. And what DOTR does, it's really a great program for all sorts of people who are working in different microbial systems. It takes all your sequences and assigns them to operational taxonomic units based on the genetic distances between the sequences. And then it looks at the frequency at which each of those OTUs are observed, and it constructs rarefaction and collectors curves for various measures of richness and diversity. And in Patrick's paper, he uses some example data sets, and one of them that he looked at was Craig Venter Sargasso Sea data set, which was generated by shotgun sequencing. And what he did was, you can see here at the 3% level, after looking at about 700 clones, they estimate there's going to be about 150, maybe 200 species in the Sargasso Sea. So this is a nice point of comparison for our own samples. And in fact, when we ran DOTR on our samples, this is just FS396, you can see that these curves are still climbing rapidly, even at this 5% level. And if we look at some of the diversity estimates that DOTR gives out, these are non-parametric estimators that, again, have become a very common tool in microbial ecology. They were actually adapted from mark, release, and recapture statistics that were used in, counting rabbits and things like that. So they consider the proportion of species that have already been observed or recaptured compared to those that are observed only once. And they appear to be quite rigorous for microbial estimates. And you can see that at this 3% level, for example, just in this diffuse vent, they estimate that in a single liter of vent fluid, there's probably over 20,000 species. And I use that term loosely because we're simply defining species by genetic distance in this case. And so what we're doing here is we're able to sample very, very deeply into the microbial world using this 454 sequencing strategy, and we're getting a much more complete picture of what organisms might be there. But really, numbers are one thing, and that's mostly what we reported in our PNAS paper, but we really want to know who the players in this ecosystem are. So we can take a look at the taxonomic breakdown. And again, I'm just going to focus on the two diffuse vent sites, since that's what I study and we have the most data from. And this is a breakdown of the taxonomy just from those two diffuse vents. This is at the 3% level and sort of at class level. And you can see, in fact, that these two samples are quite different. They have a lot of the same colors, but the relative abundance of the colors are different. And in fact, this sample is dominated by these epsilon proteobacteria, whereas this one has more gamma proteobacteria. And when I first saw this, I immediately went and looked at the chemistry from these two vents, and in fact, it's quite different. There are a number of indicators here that suggest that this vent over here, FS396, has a much higher carbon dioxide content. And in fact, this vent was effervescent or bubbling when these samples were taken. And that's shown with this elevated hydrogen sulfide to temper ratio, the suppressed pH, the higher alkalinity, and also the higher iron. And this is a characteristic of a lot of vents that we see at Axial that have a high CO2 content, a low pH, and an elevated alkalinity. So what we're seeing here is a possible link between the different microbial composition and the 454 sequences that we're getting back. We can also just look at one aspect, one group, and look at the diversity within. And so in this example, we're looking at the epsilon proteobacteria. And this is just, again, at that 3% level. And what you can see is there's a huge amount of diversity just within this one group of organisms. Now, epsilon proteobacteria are known to be key players at hydrothermal vents. They were only recently cultured really in the last 3 or 4 years. They're very phylogenetically, physiologically diverse, and they can be both mesophilic and thermophilic. They can use a variety of electron acceptors and donors from both seawater and vent fluids, allowing them to exploit both of those gradients. And many of them are autotrophic and fix carbon dioxide using the reverse TCA cycle. But what's especially interesting about this sort of data set is this pattern here, where a few tags dominate the samples, but in fact the bulk of the diversity is made up by these very low abundant tags. And I'd like to illustrate that point in our next few slides. So this plot is a little bit confusing, and so I'm going to walk you through it. So this plot basically shows the similarity of all of our 454 tag sequences from all of the data to the V6 reference database. So this red curve shows the distribution of all the tags to their percent difference from their best match in the reference database. This blue line is cumulative and shows the percent of our tags and how different they are from the best match in our database. And finally, this green tag, which shows the percent of the unique reads and their distance from their best match in the database. So if we just take a look at this blue line, the cumulative curve, you can see that about 25% of our tags are dead-on hits to something in the database. And if we keep climbing up that curve, about 40% of them are within 3% of something in our reference database. And in fact, over 75% of them are within 10% of something in the database. So this basically means we're covering the known universe very well using this sequencing strategy. But if we take a look at this green line, the percent of the unique reads, we see that only 20% of our tags are within 10% of something in the database. And in fact, the bulk of these unique tags, over 80% of them, are very different from anything we've seen before and are quite divergent. And it's really these very divergent groups that also occur at low abundance, as indicated by this red line, where you can see that these very different sequences occur at low abundance. This is what we're terming the rare biosphere in the deep sea. We can look at this another way by looking again at rarefaction curves just from sample FS396. And if we compare sequences based on how distant they are from the reference database, you get 4 very different curves. And this is sort of what the Sargasso Sea data set looked like, right, where it's flattening out at a relatively low number. And these sequences are very close to things we already know about in the database. But as you get increasingly divergent sequences, you get increasing estimates of diversity in the deep sea. So this concept of a rare biosphere isn't a new one, but we now feel like we have a tool to regularly measure it in the environment. And we're trying really to understand its significance, and now possibly will be able to. So the rare biosphere may simply reflect biogeography and many yet to be discovered habitats. So what's rare in one habitat might be quite common in another. And these rare organisms might also be a source of genomic innovation. And this can help us understand how microbial communities seem to recover from all sorts of environmental catastrophes. And also it might help us explain the genetic novelty that we find in almost every genome and metagenome sequence to date. The extreme phylogenetic diversity of the rare biosphere suggests that it's been around for a long time, possibly over geological timescales, and might have had a very important role in shaping planetary processes. And finally, low abundant populations at one site might become dominant in response to environmental change. And in that regard, the rare biosphere may serve as sentinel for global change. So a recent paper in, I don't think that came out on anybody's slide. It didn't come out on mine. But a recent paper in Trends in Microbiology tried to illustrate this. And what you can see, this on the y-axis here is the number, and this is the taxon rank. And this red part of the curve are those abundant organisms. And this long tail here are the rare organisms. And in this paper, Pedros Alio says that we've really only been able to detect these abundant organisms with current molecular methods, and every once in a while with culturing, we pick up some of these rare organisms. And we believe with 454 sequencing, we're now able to get the whole look at all of this biodiversity. And so what I'd like to do really for the rest of my talk is what Carl alluded to in the beginning. And I'd like to go through some examples of sites we might want to look at or situations we might want to look at and apply this technology not only in regard to the rare biosphere, but also in microbial populations that are going to be changing or responding to different environmental shifts. And I want to make this especially applicable to the science going on not only in the astrobiology community, but also NASA in general. So we can take a very brief look at the history of life on our own planet, as shown here. And you can imagine major events in our history that really impacted life. And I've obviously just highlighted a couple of them that we can think about, including the buildup of atmospheric oxygen and the origins of multicellularity. But obviously there are more specific examples, and one of these includes Snowball Earth. As we know, Snowball Earth hypothesis is based on geological evidence from multiple glaciations at sea level at low latitudes. And really the leading explanation for these snowballs is a runaway ice albedo effect. And basically we're putting so much solar energy back into space that we freeze over. Obviously, life was not destroyed by these processes. It was able to continue just fine, but we really have to think about how it might have changed how evolution happens, how organisms interact and respond to such a dramatic change globally. And this is some work by Kirschvink et al., where they looked at sort of a post-Snowball Earth setting and what might have been happening, and it's just illustrated here. So upon initial melting, a cyanobacterial bloom may occur, and then you have all this oxygen getting back into the ocean, where what used to be an anoxic, metalliferous ocean is now reacting with this oxygen, and you can imagine that there are both manganese oxidizing bacteria, iron oxidizing bacteria, other processes that are going to be mediated by microbes. And so you can imagine that both at the onset, during, and after a Snowball Earth, there are particular responses in the microbial communities to these different shifts. Of course, we have modern-day phytoplankton blooms, which are of great interest to a lot of people, especially in oceanography. This is a picture of a phytoplankton bloom in the Arabian Sea. This is with the ocean chlorophyll concentrations. The Arabian Sea has a very small window of time when phytoplankton can occur, and it has to do with the monsoon seasons and the winds coming in from the southwest, which allows water to upwell from the cold, nutrient-rich water to upwell from the deep. And it also brings in nutrients from land. And so these added nutrients feed the phytoplankton bloom. And in response to phytoplankton blooms, we also often see a response like this. And here on the y-axis are just the number of bacterial cells, and here we have chlorophyll. And this is presumably due to heterotrophic bacteria, which bloom in response to the increase of DOC, or dissolved organic carbon, which is being leaked out of algal cells. But in fact, we don't know that much about how diversity changes in response to phytoplankton blooms, and we think this is now a great environment to apply our 454 technology to. In addition, just this last summer, we know we have things like dead zones and toxic algae blooms, and so you can imagine we're just beginning to understand what causes those toxic algae blooms and other human sort of impacted environments, and we can begin to follow the microbial response. Here's another example of a marine environment that experienced both dramatic and spatial and temporal shifts, and that is deep sea hydrothermal vents. This is an illustration of a diking eruptive event occurring at a mid-ocean ridge, where you have a dike extrusion that results in eruption, a lava eruption on the seafloor. And this causes very extreme changes in heat flux, in chemistry, and in porosity and permeability of the crust. And there have been some studies that have looked at the response of the microbial system to this type of event, and that's shown in the schematic here. And you can imagine sort of a steady state system that sets up over longer periods of time with perhaps some deeper organisms that are living in the crust, kind of just barely eking it out on those few hydrothermal nutrients that are around. But in fact, there's organisms that are living off of seawater, maybe some of those epsilons that are making a good living. But when you have this influx of hydrothermal nutrients and heat, these isotherms get pushed up, and organisms that can take advantage of that situation are going to bloom. And that's what's shown here. And in fact, you might even have a secondary bloom then of perhaps even seawater organisms, heterotrophic aerobes, that can then feed on that primary bloom. And eventually you're going to settle down to some new steady state. Maybe those organisms that were just barely making it by now are dominant, or vice versa. We can also think about, well, I don't know if this was supposed to be a picture of Hurricane Katrina. It's not on my slide. But we can also think about natural disasters such as Hurricane Katrina and its aftermath. And in response to Hurricane Katrina, a number of microbial ecologists traveled to New Orleans to look at Lake Pontchartrain, look at the canals, and try to evaluate whether or not this water was really toxic, as was being put out in the news media. And here we can see before the hurricane and after the hurricane and where the flooding occurred. Linda Amaral-Zettler here at the MBL was one of those scientists who went to New Orleans to get follow-up samples, and she's particularly interested in looking for potentially pathogenic microbes. And some of her data is shown here on the right. And these big blue chunks are basically marine or freshwater bacteria, and these small yellow slices are microbes that she believes are potentially pathogenic or related to pathogenic microbes. And so from this data set, these look like low abundant organisms that maybe did bloom right after the hurricane, but it doesn't look like they are now. But the fact that they're there suggests that under the right conditions, perhaps something more dangerous could happen. And I think it emphasizes the need to monitor these types of systems so we don't just respond when something really terrible happens. Of course, we have a very big human disaster lurking on the horizon, actively occurring, with increasing CO2 levels and temperatures on our planet. And we don't really know what the microbial response is going to be. We know that this will impact CO2 solubility in the ocean. It's certainly going to affect the biological pump. But we don't know how microbial communities are going to respond. And so it's essential that we begin some sort of census so we can track and follow these populations over time. We can also think of another human-impacted area where we need to be able to assess and track microbial communities and where low abundant organisms might in fact be very important, and that's with planetary protection. So you can imagine applying this type of technology to make sure you know exactly what's going into space and then what comes out. And we are in fact looking into collaborations with folks at both JPL and NASA to apply this technology for screening space materia for potential contaminants. We can also think about life outside Earth, such as Mars. I sat in on a workshop last fall for the Microbial Scientist Exploration Initiative, and we talked a lot about sort of changes on Mars and Europa and how this might impact microbial populations. And so I took from some of my notes some of the ideas that people had there, which I thought were really interesting. We know that Mars has what's called a chaotic obliquity, and that's shown here, where it moves from a low mean obliquity period through a transition to a high mean obliquity period. And what this means is heating, there's differential heating in the polar regions. And this can cause a total redistribution of ice and water vapor on the planet. And so we can see there might be important linkages between the solar forcing, how the climate responds, and geological consequences on Mars. And we might want to think about, for example, how microbial populations could respond to those changes as well. This is just an illustration of the ice evolution on Mars, and this angle between the white arrow and the dotted line denotes that Martian obliquity. And so you could think about, well, is the Martian biota in some sort of dormant stage when this obliquity winter, and what might happen when the obliquity changes and there's much less ice? Would they hold on to some sort of genomic information? What are the time constraints on that persistence? We also know that Europa is a very dynamic system. It has an extremely eccentric orbit, which is shown here. And it also has a complex geology. And it's believed that the eccentric orbit, the geology, and these tidal heating are all tied together. And in fact, this is a model looking at Europa's ice thickness as it varies based on changes in both orbit and tidal heating. At the workshop I was at, we also talked about how the possibility of a convection occurring in the ice shell of Europa, and also what I thought was really fascinating, this idea that in Europa's very early stages, it could have had a completely open ocean. And so again, this is another dynamic environment. When we think about life on Europa, what we might want to try to detect, we have to think about all the stages that this planet or this moon might have gone through. So in conclusion, we believe that this 454 tag sequencing approach provides an in-depth initial view on total diversity of microbes in an environment. We think it's very efficient and it's high throughput, which allows for intensive sampling of all sites of interest. It can detect both major and minor population members, and it offers a really neat tool to fingerprint microbial communities over time and space for correlations with biogeochemical activity. You can imagine doing a biogeography experiment as well. We believe it's an important complement to metagenomic culturing and in situ hybridization investigations. And we're going to expand it to include the archaea and the eukarya. We're in discussion about whether or not it's going to work for viruses. And we recently received funding from the Keck Foundation to begin our census and apply this in a variety of oceanic environments. Our GS20 from 454 was delivered about 3 weeks ago, so we're just getting things up and running here. But we already have samples in the pipeline with collaborators all over, and that includes more samples from these complete cruise track lines from the North Atlantic. We're collaborating with people at the University of Hawaii to look at the Hawaii Ocean Time Series at Station Aloha. This is one of the best long-term data sets we have in the ocean. We're going to be working more on human-impacted areas through the Woods Hole Center for Oceans and Human Health. That includes collaborators in Woods Hole as well as in MIT. And we're also working with colleagues through the Ocean Drilling Program and the Ridge 2000 program to look at both subsea floor basalts and subsea floor sediments. And of course, we're going to be looking at, in my favorite environment, and that's deep sea hydrothermal vents, in this case of the Pacific Ocean. And because I think I have a little bit of time, I'm going to show you a video from a very dynamic environment that you can imagine organisms responding to quite rapidly. And that is from this site here in the Western Pacific. This is a cruise I participated in with NOAA Ocean Explorer this last spring. And I'm going to show you a video of this active eruption taking place on the seafloor at about 500 meters depth. And until last spring, this is something that had never been seen before. Let me see if I can do this right. So while you're watching, you should look for gas bubbling, you should look for sulfur, you should look for lava bombs, and especially look for a red flash, and that's actually a magma extrusion, and that also is the first time it's been seen on the seafloor. So I'm just going to wait a few more seconds and let this finish loading up. I should mention that no people were harmed in the filming of this eruption. This was a remotely operated vehicle, Jason 2, which is operated out here in Woods Hole. So, and if you could hear this, we actually have hydrophone data. There's the red flash. We actually have hydrophone data that's tracking, that's listening as we're viewing this eruption. And we actually got samples in 2004 from this site.
We're not going to see it.
It'll take us off.
We actually got samples.
See it, we can request it from her. She can email it.
It's available on the
Hey,
NOAA site.
Steve, you all right? Your mic is open.
And so we actually got samples from this site in 2004 before it started going crazy. And then in 2006, we spent about a week watching this volcano go through its full eruptive cycle. So we have samples from when it was just a wisp of 20-degree fluid up to when it was full bang, about 150-degree Celsius eruption occurring. So this is just an extreme example of the type of environment that we're going to be studying with the 454 sequencing technology. And this collection of samples from all over the ocean is really going to allow us to explore some exciting things, I think, in the microbial world, in the sea, and potentially in other environments as well. So just in conclusion, I want to acknowledge our collaborators in the Netherlands, Gerhard Handel and Jesus Arietta, for providing those Transat samples. We have a lot more of them in our freezer to get working on. The Alfred P. Sloan Foundation, which has funded all of our pilot studies. The Astrobiology Institute that funds me. The Keck Foundation, which is helping us with our purchase of our 454 sequencing machine and the beginning of our ICOM initiative. And if you want to learn more, you can check out the paper Carl referred to, or you can just drop me an email. So with that, I'll take any questions.
Thank you, Julie. Well done. Okay, so just to review again, because we have a lot of sites connected, please raise your hand in the chat area, or please raise your hand in the participant list, or put a note in the chat area to Marco Bolt, which is actually Marco Bolt and myself today, and we will call on your sites. So I don't see anything right now, but so now I'll just open it up. Any questions? Oh, wait, hold on, sorry. University of Rhode Island. You can now open your mic and talk officially. We're not hearing you.
How's that?
Great. You're dark, though. If you can turn the light. Yeah, that's good. We can hear you.
You don't know how only one gets into this cell.
Yeah, on the 454 technology, can you tell me how only one one strand of DNA gets incorporated on the bead that actually has multiple receptors on it?
Right. So you have 4 possibilities that could happen, and they select just for, well, there's 2 steps. They select just for beads that only have one adapter picked up on them, but they go through a selection process to try to wean it down. But in fact, you can get false reads that have more than one amplicon on them, and the software is built to recognize that. Your sequencing looks really, really messy because you can see multiple incorporations in one flogram. And so it really happens a little bit in the lab work part, but mostly in the bioinformatic part.
And then you try and control that by how much DNA you add into the ratio of beads?
Yeah, so there's,
okay,
you do a dilution series with your beads and your amplicons and those sorts of things.
Right. Thanks, Julie.
Yep.
Great. Thanks, David. University of Arizona. Go ahead.
Nick Wolf here. Very nice talk, Julie. Coming at this from outside of biology, I'm puzzled by the following question. Since we know that the same ribosomal RNA can be associated with different metabolic processes, how do we know that the reverse can't happen, that what you're seeing are essentially the same microbes with slightly different ribosomal RNA?
That's a very good question, and the best tool we have to look at that right now is our reference database. And so if we go through our entire reference database and have 120,000 unique sequences, we can only pull out 44,000 unique V6 regions. And so we already see a little bit of a disparity there. And so what we're trying to do is look at the diversity at a level that we think is meaningful, and that's why we're not just looking at individual sequences, and we're using this, this distance approach where we collapse things into 3%, 5% levels, so that we can hopefully get rid of a little bit of that wiggle room. But actually, this tells us nothing about function, right? We have no idea if these 20,000 estimated microbes are all doing different things or if they're all doing the exact same thing. This is a first step in getting closer to a census. And what we're trying to come up with are creative ways then to go back into the environment and study those organisms to find out what they're actually doing in the community.
Thank you.
Thanks, Nick. Okay, MSU, are you there?
So my question is, this tail end, this is very popular, and we have seen it in soil a lot, tail end of microbial communities where you see a lot of very unique OTUs. How do you get a better characterization of that now that 454 is available?
Right. So the first thing we want to do is our hope, well, one of our hopes is that by doing a bunch of different samples from a bunch of different environments, we might be able to see some patterns, or at least find places where those rare in one sample occur in another sample. So begin to build a picture of these divergent OTUs instead of just one researcher seeing them with their particular PCR approach and then no one ever seeing them again. So the idea is if we treat all the samples equally, maybe we'll be able to find them. The second step, and something we're trying to show in a proof of concept, is to design a primer based on that unique V6 sequence and put it with a universal bacterial sequence, go back into your sample, try to amplify out more of the genomic DNA, the 16S DNA in this case, possibly screen phosmid libraries for this gene, try to visualize it. We're trying to get to that point. But the first step is really to see how rare is it actually, or is it just that we haven't been looking enough?
Well, thank you very much. And if I could ask another question, it will be, what if you actually want to apply it to specific functions, genes related to specific functions? How would you change your approach?
Well, right now 454 is really limited to about 100 base pairs. So any primer design you could think of where you're going to get enough information out of about 100 base pairs, go for it. You should give it a shot. The new generation of machines, they say, are going to get about 200 base pairs, so maybe that gives us a little more leadway in the types of genes we could design primers for. But that's the constraint right now.
Thank you very much.
Okay, University of Washington. Are you guys there?
Hi, it's Tom Quinn here. Actually, my question was very similar to Nick's, and that is, how can you tell whether you've actually got diversity in phenotype, since you're just measuring diversity in genotype? So just to push you a little further on that, what ideas do you have to try to figure out the diversity of function? Is it possible to get a hint of protein structure from your sequences, or are you going to have to do more sort of whole cell culturing and things like that?
Yes. Using this technology, we're not going to get any sense of function. We're just going to get binning into taxonomic identities. We are, though, going to get fingerprints for microbial communities in all sorts of different environments. Maybe we'll be able to pick up patterns. We only find certain tags in oxygen deplete zones or in high temperature zones, for example. And that's how microbial ecology has been going for the last 10 years. And in the last few years, people have started using very creative culturing techniques to get a sense, to find out what those organisms are actually doing. But really, I think the key is going to be a combination of culturing and doing some more functional genomic methods to get a true handle on that. This is just a cataloging of diversity. That's all it is.
Thanks, Tom. Anything else from Washington?
That's it here. Thank you, Julie.
Thanks.
Yep.
Okay, Goddard Space Flight Center. You have a question there?
Yeah, hi, it's Mike Wuma. I'm interested in the potential applicability to systems that might be related to Mars or subterranean life on Mars. I wondered if you had any plans to go to the Lost City Formation, for example, other type of community and so forth.
Well, we don't have any plans to, but we certainly know about a lot of the data that's being generated from Lost City and other ecosystems driven by serpentinization. We know that at Lost City, actually, the archaeal community is very simple. We made up only a couple different organisms, but the bacterial story looks a little bit more complicated, and actually I think the application of 454 to that environment would be really interesting.
Thank you.
So if anyone has samples, send them along.
Thanks, Mike. Okay, I don't see any more hands raised. I just want to give a chance to the people who came in on the conference call number to ask any questions. Any questions there from the telephone? And Mike, it should be unmuted. Good. Okay, great. Well, thank you all very much for attending, and again, thanks to the Johnson Center for the use of their video conferencing facilities. I think it worked wonderfully, and I was very glad to have ISDN connection today. Thanks for everyone here, and welcome to Carl Pilcher. Julie, have a good time at sea, and we'll see you all next month. Bye.
Thanks everyone.
See ya.